Assalamualaikum and very good morning. We are from Group 1 who would like to share about erythropoiesis. What is erythropoiesis? Erythropoiesis is the production process of red blood cells. Erythropoiesis takes place in the bone marrow where hematopoietic stem cells differentiate and eventually shed their nuclei to become reticulocytes. It is triggered by erythropoietin. Erythropoietin is a hormone produced by the kidney with small amount made by the liver. Did you guys know, in order to produce a normal red blood cell, our body needs protein, iron and copper, folic acid, and vitamin B6 and B12. Protein is important to transport oxygen from lungs to tissue and transport carbon dioxide from tissue to lungs. 70% of body iron is found in red blood cells. It is because iron is responsible for hemoglobin production. Folic acid and vitamin B12 required for DNA synthesis and important for the formation of new erythrocytes. Erythropoiesis is one of the important processes in our body. Erythropoiesis helps our body maintain sufficient oxygen delivery to tissue so that body can bring carbon dioxide as a waste product to lungs. Hemoglobin is an important protein in the red blood cell that carry oxygen from the lungs to all parts of our body. Indirectly, erythropoiesis will enhance synthesis of hemoglobin in the red blood cell. Hemoglobin also gives the red color of our blood. Erythropoiesis also keep the red blood cell count balanced. Body can be affected if the red blood cell count is too low or too high from the normal range. Last but not least, with the help of vitamin B12 and folic acid, erythropoiesis can hasten maturation RBC. Lack of either one can cause maturation failure in the process of erythropoiesis, which manifests clinically as reticulocytopenia, an abnormally low amount of reticulocytes.
We all know it is normal for all living cells to undergo its own life cycle. So does the erythrocyte itself. The development of the red blood cells are a cell synthesis process under hematopoiesis. These double concave cells are being produced in different organs according to different stages of an age of an individual. For an adult, the red blood cells are being produced in the vertebrae, ribs, sternum, and other essential appendage bones. Erythropoiesis begins with the stem cells named as hemocytoblasts, and then subsequently followed by the progenitor cells named as proerythroblasts. Fundamentally, stem cells are pluripotent, which means they can turn into any types of cells, but the differentiation of themselves are not specific. However, for the progenitor cells, they can directly differentiate and turn into a specific types of cells.
Still confused? It's okay. Let's try to imagine the hemocytoblasts as the paddy and the proerythroblasts as well-cooked rice. So, from here, you can see how the paddy is pluripotent because it can be processed either into rice or wet powder or processed rice cake or oat bars and many more. However, if we cook the rice, they can be modified directly into a wide variety of dishes such as nasi lemak, nasi kerabu, pulut nasi, bubur nasi and many more. Just name it. So, this is the simplest way on how to compare the pluripotent and the multipotent. Back to the process. With the support of the microenvironment of bone marrow, hemocytoblasts will differentiate themselves forming two proerythroblasts. Proerythroblasts are equipped with an immature nuclear chromatin. They also present a basophilic cytoplasm. Then, they will differentiate into basophilic erythroblasts. In this state, the chromatin starts to condense together and the basophilic cytoplasm is reaching its maximum limit. Basophilic erythroblasts will proliferate into polychromatic erythroblasts. In this state, hemoglobin starts to be produced, thus leading to an eosinophilic cytoplasm. On the other hand, the chromatin already clumped together, which indicates the maturity of the nuclear chromatin. This will be preparing the nucleus to be expelled in the next stage. Polychromatic erythroblasts will differentiate into orthochromatic erythroblasts. In this stage, the hemoglobin starts to accumulate in order to prepare the reddish cytolytic for the next stage. The cells also start to be pikinetic. Orthochromatic erythroblasts will then expel the matured nucleus from the cell in order to transform into reticulocytes. In reticulocytes, they are still contain some RNAs, so usually it takes 1 to 2 days for it to be dissolved. Only after this, the reticulocyte is officially turned into a mature erythrocyte. So, here are the proper steps. So, throughout the process which took place for only a week, we can conclude that erythropoiesis involves the decrease of the cell size, which is gradually for each level. On the other hand, the maturation chromatin leads to the assembly of the hemoglobin in the late erythroblasts. Apart from that, the maturation of the nucleus is also being stimulated, which is fundamentally essential as the final step in the erythropoiesis.
However, it is still important to know that for a certain time, the terms normoblast will be used. But don't worry, because naturally, normoblasts can be defined as the nucleated normal cells occurring in the red marrow as a stage of the development of the red blood cells. So, literally, normoblasts are referring to the erythroblasts.
As the previous presenter had already explained the steps and how a hemocytoblast differentiate and proliferate itself in order to become a mature erythrocyte. Now we will be looking closely into each one of them in order for us to further understand its morphology and its characteristic. There are 7 cells that we'll need to familiarize ourselves with and to know which is which. So let's begin to see one by one of them. So let's begin. First up in the team would be the hematopoietic stem cell. It is an immature cell that can develop into all types of blood cells, including the white blood cells, the red blood cells, and the platelet. These cells are found in the peripheral blood and the bone marrow. We can also call them as hemocytoblasts. The hematopoietic tissue contains cells with long-term and short-term regeneration capacities, enabling them to give rise to multiple types of cells. They are round, non-adherent, with a rounded nucleus and low cytoplasm to nucleus ratio. As you can see from the picture, the cells are round and they have a very big nucleus. The nucleus is so big that the cytoplasm looks so small, not really small, like secret compared to the nucleus, so they have a low cytoplasm to nucleus ratio. Hemocytoblasts will constantly divide into 2 different kind of cell lineage. The first one will be the myeloid cells, and the second one will be the lymphoid. As our topic today is about erythropoiesis, we will be focusing more on the myeloid cells, as this cell will then differentiate and become the erythrocyte. Myeloid cells, with the help of erythropoietin, which will be discussed later in the video, will then develop into pro-erythroblasts. We are now in the second stage of erythropoiesis. Their proliferative capacity is more limited compared to the preceding stage, which is the hemocytoblast. There are several nucleoli, which are the small basophilic spherical bodies located in the nucleus, and it occupies most of the cell volume. The chromatins consist of a network of fine red-pink strands. The most distinguished feature of pro-erythroblast to its corresponding myoblast is that it carries more basophilic peripheral cytoplasm. As you can see from the picture, the cell actually doesn't look much different from the hemocytoblast, but then you can see the nucleus should be more pink as it has fine red pink strands, and should look a little bit more purple or bluish due to its basophilic peripheral cytoplasm. In this stage, proerythroblast loses its nucleolus after its hemoglobin genes start producing tons of RNA, for example the mRNA and the tRNA. And this is the start of basophilic erythroblast. The cell is much smaller than the original blast and has an intensely basophilic cytoplasm due to the accumulation of ribosomes. The ribosomes are the one that's synthesizing those RNAs and produce the blue light colors, which is why the cell looks quite bluish. As you can see from the photo right here, you can get a sense of the differences between proerythroblast and the basophilic erythroblast. The main difference would be the size. As you can see again, the basophilic looks much smaller than the proerythroblast. The color looks quite similar, but the basophilic shoot looks more bluer and more purplish compared to the proerythroblast due to the accumulation of ribosomes. Alright, so in this stage, and the previous stage as well, RNA is converted into protein by ribosomes. The protein is hemoglobin, which is why it gives the red color to the cell. However, at this stage, not all RNA are able to be converted or translated as hemoglobins. There are still some blue colors, or the cells still having a slightly basophilic color. These cells have a larger diameter than the mature red blood cells. Having both basophilic and hemoglobin colors, this cell is named as polychromatic, which means multicolored. They have both the blue and the red. Again from the photo, in order for you to differentiate between the previous cell and the polychromatophilic erythroblast, is that it became again much smaller than the basophilic, and the nucleus actually looks more darker than the basophilic due to the hemoglobin releasing the red color, giving the red color to the cell. The RNA continued being translated by the ribosome, causing the nucleus to be more condensed than before, and this caused the nucleus now appears much smaller. Normal blast should have more hemoglobin than RNA, and it looks more purple than the polychromatophilic erythroblast. Some may also call this stage as orthochromatic erythroblast. At the end of this stage, the cell ejected its nucleus and some of its organelle, for example the mitochondria. Compared to the previous stage, which is the polychromatophilic erythroblast, this normal blast should look more purplish compared to the previous stage because of the hemoglobin now releasing more red colors. And also the nucleus looks much smaller right now. From the previously normal blast, when the cell ejected its nucleus and its organelle, the mitochondria, so now the cell is called as reticulocytes. The reticulocytes develop and get pushed out from the bone marrow. It then circulates for about a day in the bloodstream before developing into a mature red blood cells, which are the erythrocytes that we're looking for. Reticulocytes appear slightly bluer than the mature red blood cells when stained. That's how we differentiate between both of them. These are the pictures of blood cells being stained, and as you can see, when there are quite bluish strands in the cell, that will be the reticulocytes, and the clear looking cells are the mature erythrocytes. Lastly, we arrive at the last part of erythropoiesis, which is the mature red blood cells or the erythrocytes. The erythrocytes, like a cell nucleus, it is deformable, flexible, able to adhere to other cells, and are able to interface with the immune cells. The human erythrocytes are biconcave shaped, about 7 to 8 micrometer in diameter, and has about average lifespan of about 100 to 120 days. Deviations in morphology in terms of size, of the shape, or the color, or the distribution may associated or perhaps diagnostic of disease entities. Yes, normal red blood cell should look like this, but when it has several abnormalities like these, then it might be a sign of disease. There are a few examples here. For example, the macrocytic RBC, which the RBC looks bigger than what it's supposed to look like. It may associated with the vitamin B12 deficiency, the liver diseases, or maybe they have like difference in terms of its shape. For example, the sickle cell RBC. It may associated with the hemoglobin SS disease, the hemoglobin SC disease, and more.
Hello, my name is Masara binti Abu Bakar, and I'm going to explain about the growth factor. The growth factor is a naturally occurring substance capable of stimulating cell proliferation or growth, wound healing, and occasionally cellular differentiation. These growth factors are usually made up of protein, such as cytokines. So there are several growth factors that are involved in erythropoiesis, which are erythropoietin or EPO, insulin growth factor or IFG1, interleukin-3, granulocyte macrophage colony stimulating factor or GM-CSF, granulocyte colony stimulating factor or GCSF, and stem cell factor or SCF. Each of these growth factors has its own function in erythropoiesis and which cell will it affect. Let's start with erythropoietin. Erythropoietin or EPO acts on red blood cells to protect them against destruction and stimulates stem cells of the bone marrow to increase the production of red blood cells. Next is interleukin-3. Interleukin-3 induces the proliferation and differentiation in both early pluripotent stem cells and committed progenitors. There are also other interleukins such as interleukin-9 and interleukin-6 which helps in differentiating the cells. Stem cell factor or SCF promotes cell survival, proliferation, differentiation, adhesion, and functional activation of cells at multiple levels of the hematopoietic hierarchy. In other words, it regulates the hematopoietic stem cell activity in the bone marrow. Next is granulocyte macrophage colony stimulating factor or GM-CSF. GM-CSF promotes myeloid cell development and maturation, while granulocyte colony stimulating factor GCSF stimulates the bone marrow to produce granulocytes and stem cells and release them into the bloodstream. Last but not least is insulin factor growth or IFG1. It is the major mediator of growth hormone GH and stimulated somatic growth and inhibits apoptosis in hematopoietic progenitor cells. Based on the diagram, interleukin-3 will proliferate and differentiate the hematopoietic stem cell or HSC into CFU-GEMM. CFU-GEMM is a colony-forming unit that generates myeloid cells. CFU-GEMM cells are the oligopotential progenitor cells for myeloid cells. They are thus also called as common myeloid progenitor cells or myeloid stem cells. GEMM stands for granulocyte, erythrocytes, monocytes, and megakaryocytes. To make hematopoietic stem cells differentiate and grow into pro-erythroblasts, CFU-GEMM must give rise to CFUE, which will lead to the formation of pro-erythroblasts. CFU-GEMM then will differentiate to BFUE early or bursting forming unit erythroid, which is the earliest precursor cell specific to erythroid lineage, by interleukin 3, GM-CSF, and GCSF. BFUE early then continues to develop into BFUE late with interleukin 3, GM-CSF, and IGF-1. After BFUE late was developed, it will have a lower number of EPO receptors. Thus, BFUE late will differentiate into CFUE by interleukin 3, GM-CSF, and EPO. As the cells mature into erythroid colony forming unit erythroid or CFUE and proerythroblasts, it will highly dependent on EPO only. So EPO will be the major growth factor during this stage. However, as erythroblasts become more mature, it will have fewer EPO receptors and are less sensitive to EPO stimulation. Reticulocytes and erythrocytes have no EPO receptors and thus are unresponsive to EPO.
Hi, today I will be talking about regulation of erythropoiesis. I really hope that all of you guys can stay with me in this session because the explanation is quite long. When talking about regulation, usually we will focus on how our body system will respond to the changes of normal erythropoiesis activity. On this slide, the diagram shows roughly the effect of when the erythropoiesis activity is lower and higher than normal. If the activity is lower than normal, it can cause hypoxia in the blood. If erythropoiesis activity is higher than normal, it can cause polycythemia rubra vera, which I will explain it more later. The information that we have to know before we proceed with the lecture. The first one is the main regulator of erythropoiesis, which is hormone erythropoietin, EPO. The second one is the location of production, which at kidney, specifically in the renal interstitial peritubular cells. As you guys can see below here, an illustration of where is the interstitial peritubular cell. And this illustration I drew myself is a nephron, the smallest unit of kidney. In the normal condition, erythropoietin is not produced. This is because the partial pressure of oxygen demand is enough. So that means the amount of RBC in the body is normal. But the question here is, how our body decides not to produce hormone erythropoietin? When the amount of oxygen is enough, the oxygen molecule will activate the enzyme prolyl hydroxylase, which will inhibit HIF2 alpha with OH group and with molecule that specifically works to destruct the protein. And HIF2 alpha will be sent to proteinases to be destructed. If you guys are wondering what is HIF is hypoxia inducible factor. It is a protein component, which it has two types in erythropoiesis. The first one is HIF2 alpha that can be found in the cytoplasma, and the second one is HIF2 beta that can be found in the nucleus. What the functions of these two HIF? If HIF2 alpha and HIF2 beta combine, they will form a transcription factor that will stimulate the production of erythropoietin. So that's why when the oxygen level in our body is enough, the enzyme prolyl hydroxylase will inhibit HIF2 alpha to be destructed by proteinases, because we don't want to have any more erythropoietin, because it can lead to excessive amount of red blood cell in our body. So what happens if the amount of oxygen is lower than normal? In this case, low level of oxygen equal to low level of erythropoiesis activity. And this situation can form a condition we call as a reduced partial pressure. This is a stimulus that will be detected by renal interstitial peritubular cells that will surge the EPO production, hormone erythropoietin, and it will stimulate the RBC to increase in the bone marrow. So that means the hemoglobin will increase and the partial pressure oxygen back to normal. The next question that we have: how the stimulation of erythropoietin happen? When the amount of molecule oxygen is low, it cannot activate enzyme prolyl hydroxylase, which means it cannot inhibit HIF2 alpha with OH and destructive molecules. That means no destruction of HIF2 alpha occur. Free HIF2 alpha will combine with HIF2 beta in the nucleus and form transcription factor that lead to erythropoietin production. The condition of reduced partial pressure of oxygen may be due, first one, lack certain dietary such as iron, B12 and folate. The second one is kidney failure due to decrease the sensitivity of renal interstitial cells and can cause anemia. The third one, environment at high altitude such as mountain that can cause hypoxic. In the case of overproduction of red blood cell will lead to polycythemia rubra vera. It is a disorder where too many red blood cells are produced in the bone marrow. The blood will become thicker and the blood flow will be reduced. This condition can be the indicator of the first one, myeloproliferative disease. It is a slow-growing blood cancer in which the bone marrow makes too many abnormal red blood cells. The EPO production is off, but red blood cells are continuously produced. The second one is the bone marrow disorder. It is caused by JAK2 mutation and makes the red blood cell produced in large quantity.